Welcome to DNA Detectives, how genetic clues crack criminal cases. Today we'll explore how forensic scientists use DNA to identify people and solve crimes. From collecting samples at crime scenes to matching profiles in databases, DNA can reveal incredible truths. We'll also learn about famous cases, future technologies, and how you can get involved in this exciting field. DNA stands for deoxyribonucleic acid, and it's the code that makes up every living thing. It contains instructions for building and running cells. In humans, 99.9% of DNA is the same in everyone, but that 0.1% is what makes us unique. That small difference is what forensic scientists look for when solving cases. Understanding what DNA is and why it matters helps us appreciate its power in forensic investigations. DNA forensics is the use of DNA in criminal investigations. Scientists collect DNA from crime scenes and compare it to samples from suspects or databases. If there's a match, it could link someone to the crime. This powerful technique can confirm identity, solve cold cases, and clear the innocent. DNA has become one of the strongest scientific tools in modern law enforcement. DNA is made of building blocks called nucleotides, arranged in a double helix. The order of these nucleotides forms a person's genetic code. Everyone's DNA sequence is slightly different, like a fingerprint made of molecules. These differences are what scientists analyze in forensic labs. The structure of DNA helps explain why it's such a useful identifier in forensic science. DNA can be collected from blood, saliva, hair, skin cells, or even touched objects. Investigators wear gloves and use sterile tools to avoid contamination. They label and seal each sample carefully before sending it to the lab. Proper handling is key to preserving the evidence. Without careful collection, DNA samples may be damaged or contaminated. Once in the lab, the DNA must be extracted from the cells. Scientists break open the cells, separate the DNA, and purify it so it can be analyzed. This process removes everything else, like proteins or dirt, so only clean DNA remains. Pure DNA is essential for reliable testing. This step sets the foundation for accurate forensic analysis. PCR stands for polymerase chain reaction. It's a technique that makes millions of copies of a DNA sample. This is important because crime scene samples are often tiny. PCR helps amplify the DNA so scientists have enough to study and compare. It's like making a photocopy of the DNA so there's plenty to work with. Gel electrophoresis is used to sort pieces of DNA by size. The DNA is placed in a gel, and an electric current pulls the fragments through. Smaller pieces move farther than the larger ones, creating a pattern of bands. These bands act like a barcode for each person's DNA. This visual pattern helps forensic scientists match samples. DNA profiling focuses on areas of DNA that vary the most between people. One of these areas involves STRs, short sequences that repeat. Scientists count how many times these repeats occur at different locations. The combination creates a unique profile for each individual. These profiles are what investigators use to compare DNA samples. CODIS is a national database used by law enforcement to store DNA profiles. It includes DNA from convicted offenders, arrestees, and crime scene evidence. When a new DNA sample is collected, CODIS searches for matches across thousands of profiles. This helps quickly identify suspects or link crimes. CODIS has solved many cases that would otherwise have remained unsolved. It's a crucial resource in forensic investigations. Matching DNA involves comparing a crime scene sample to a suspect's profile. If they match, it suggests that person was present. However, DNA alone doesn't prove guilt. Investigators use it alongside other evidence. Forensic scientists calculate how likely it is that the match is coincidental. High probabilities strengthen the case against suspects. Courts consider DNA a powerful but not standalone proof. Sometimes investigators don't find an exact match but find a close relative DNA. This method, familial searching, looks for partial matches to relatives. It helps identify suspects indirectly through family ties. Familial searching has cracked cases previously thought unsolvable. It raises privacy and ethical concerns but is valuable in solving crimes. This technique broadens the scope of DNA forensics. DNA evidence can be preserved for years and reanalyzed with improved methods. Many cold cases, even decades old, have been solved using DNA. This has led to arrests of serial offenders long after crimes occurred. These breakthroughs bring justice and closure to families. DNA's durability and evolving technology make it invaluable in ongoing investigations. Mitochondrial DNA, or mtDNA, is inherited maternally and is present in many copies per cell. Its stability makes it useful for testing older or degraded remains. It helps identify victims in mass disasters and archaeological finds. mtDNA can provide maternal lineage information, complementing nuclear DNA analysis. This technique expands forensic possibilities beyond fresh samples. It is especially useful when nuclear DNA is not available. YSTR analysis focuses on DNA from the Y chromosome, which only males have. This is valuable in cases where male DNA is mixed with female DNA. Since all paternal relatives share similar YSTR patterns, It helps narrow suspects. However, it can't differentiate between closely related males. YSTR testing complements other forensic methods. It adds another layer to solving complex cases. DNA mixtures occur when samples contain material from several individuals. Decoding these mixtures is one of forensic science's toughest tasks. Experts use advanced software and techniques to separate profiles. Misinterpretation can lead to wrongful conclusions, so caution is key. Accurate analysis of mixtures helps solve complicated cases involving multiple people. It demonstrates the need for skilled forensic analysts. Preventing contamination is critical in forensic DNA testing. Even tiny amounts of foreign DNA can spoil results. Labs follow strict procedures, including wearing protective gear and sterilizing equipment. Maintaining the chain of custody tracks evidence from collection to analysis. Quality assurance programs ensure reliable and reproducible results. These safeguards maintain trust in forensic evidence. The use of DNA evidence comes with legal and ethical challenges. Questions arise over privacy and how stored DNA is used. There's debate about expanding DNA databases and familial searching. Different countries have diverse laws regulating DNA collection and use. Ethical considerations require balancing justice with personal freedoms. These issues continue to evolve as DNA technology advances. Forensic genealogy links DNA profiles to relatives via family tree research. Public genealogy databases enable tracing of suspects through distant relatives. This method identified the Golden State Killer after decades without leads. While powerful, it raises privacy and ethical concerns. Forensic genealogy is changing how cold cases are solved. It represents a blend of science, detective work, and public data. The Golden State Killer was responsible for numerous violent crimes over a decade. Despite extensive investigation, he remained unidentified for many years. In 2018, investigators used forensic genealogy to finally catch him. By uploading DNA evidence to a public genealogy database, they traced his family tree and identified a suspect. A direct DNA match confirmed his identity, bringing justice after decades of fear. This case revolutionized forensic science and investigative methods. Touch DNA refers to DNA left behind when someone touches an object. Even a brief contact can transfer skin cells sufficient for analysis. This evidence is valuable in cases lacking blood or other bodily fluids. Because it's easily contaminated, proper collection methods are essential. Touch DNA has solved many cases where traditional evidence was scarce. It's a powerful tool in modern forensic investigations. DNA evidence isn't limited to blood or bodily fluids. Everyday items like cigarette butts and cups can harbor DNA from saliva. Hair with the root can provide high-quality DNA samples. These unusual sources often link suspects to crime scenes unexpectedly. Forensic teams are trained to recognize and collect such evidence carefully. These findings have expanded possibilities for solving crimes. DNA evidence is not only used to convict the guilty, but also to free the innocent. Many individuals wrongfully convicted have been exonerated through post-conviction DNA testing. The Innocence Project and similar organizations work to identify such cases. Success depends on preserved evidence and careful testing. These cases highlight the importance of DNA in ensuring justice and fairness. They have changed the criminal justice landscape worldwide. DNA analysis plays a critical role in identifying victims after mass disasters. Families provide reference samples to help match remains. This process aids in returning remains to families and providing closure. DNA testing helps identify remains even years after disaster. It is a crucial tool for humanitarian and forensic responses worldwide. These efforts combine science and compassion. Test your DNA knowledge. First question, what does PCR do in DNA analysis? PCR is a technique that copies DNA to create millions of copies from a tiny sample. Second question, what part of DNA is used in profiling? STRs, or short tandem repeats, are the parts of DNA used to create unique profiles for individuals. What does CODIS stand for? CODIS stands for Combined DNA Index System. Fourth question, how is mitochondrial DNA different from nuclear DNA? Mitochondrial DNA differs from nuclear DNA as it is inherited maternally and is more stable over time. Fifth question, what is forensic genealogy used for? Forensic genealogy uses DNA in family tree research to find suspects by tracing relatives. How do you do? DNA science is fascinating and powerful. Thank you.